Thanks a lot. I'm sorry I couldn't be there in person, but yeah, I'm visiting California in January, so I'll make sure I'll go visit John as well. Thanks.
We sure appreciated you coming to join us in any way that you could. Thank you so much. Let's give a hand to Vivek. Thank you so much. And then our next talk is Mariam Waterman, University of California in Irvine, who's going to give more perspective on the tumor microenvironment and presumably some of the immunology there. Mariam, are you ready for your talk?
I am. Can you hear me?
We can. I'm going to go ahead and turn it over to you. And I think they are switching the camera view over here for us.
Oh, okay. All right. Thank you, Paul and Min, and everyone for the invitation to join and talk about this talk. It won't be about too much immunology, Paul. It's really going to be focused on cancer stem cells as I triaged and trimmed down my talk to make it in time. I really would have loved to have joined you in Canada, but ironically, the reason I can't join you is that I am moving to Canada. Movers come in 2 days. So in any case, let me get started. Let's see. What I want to talk to you about is a collaboration, a longstanding collaboration I've had with John and his group. I'm going to focus on some data from 2 students in my group, Lindsay Hosahama and George Chen, but also a student, Mary Lee, that was a graduate student with John. All of them have now graduated and they are in their postdocs or other careers. And this started as a collaboration when John and I were program co-leaders of a group in the Cancer Center. And as a demonstration that a cancer biologist could team up with a mathematician, we began to look for ways that we could collaborate. And I'll show you that data, the data that triggered our collaboration. But here's what I really thought of John when I first met him. It took me 6 months to understand some of his lexicon. But I want to wish you happy birthday, John. So let's put on a party hat. He's very bright, smart, and with me, patient and kind in helping me sort of get up to speed on this collaboration. John's input has greatly influenced what I think about cancer and enhanced my understanding of cancer therapies. So I hope to get to that point at the end of the talk. To introduce this talk, I want to go over 2 things, a signal transduction pathway, and then also some stem cells in the intestine. This is going to be a talk focused on the colon. So Wnt signaling, perhaps you are familiar with this pathway, but it's a signaling transduction pathway. I don't know, can you see my arrow? Can you guys see me moving this around?
Yes, we can.
Okay, thanks, Paul. A secreted protein ligand binds to receptors that turn off the activities of the destruction complex, allowing beta-catenin to go in the nucleus and turn on cohorts of genes. This is an extremely important pathway in many settings. The most important, I would say, is in stem cells during development when they are in the process of creating tissues, in adults in homeostasis, at sites where there are stem cell niches that need to replenish tissues such as skin, bone, intestine, et cetera, et cetera. And then also in injury, Wnt has a very important role to play in helping tissues repair and regenerate. And these cohorts of genes tell cells how to behave, such as grow, to proliferate, but also in stem cells to self-renew, et cetera. And they also, in this cohort I'm showing here in Wnt, have feedback inhibitors, and this will become important in the talk. The feedback inhibitor we'll talk about are the DKKs. These are secreted proteins that bind to Wnt and kind of interfere with their ability to bind to receptors. They also sort of mask this green squiggly line that I've shown here, which is a lipid tail. Wnt are actually very, very sticky to the cell surface, and so their diffusion length isn't very far. It's maybe 1 cell. When DKKs bind to Wnt and interfere with receptor binding, they mask this. And so ironically or paradoxically, when DKKs bind and interfere, they also enable longer range diffusion of Wnt, and this will become important later in the math models. I also want to mention secreted frizzled-related proteins. They're also secreted. They also bind to Wnt and extend the range, but they're not known to be a Wnt target gene. The last thing I'll say about Wnt signaling, and you probably know this too, is that at least for colorectal cancer, 80 to 85% of patients with a colon tumor, those tumor cells have mutations in genes that encode components of the destruction complex. And that just turns off and destroys the destruction complex, making beta-catenin a very abundant protein. It's always in the nucleus at high concentrations. It's always turning on these gene programs. And it's this pressure, especially for the proliferation program, that transforms cells and turns them into cancer cells. So the origin of colorectal cancer sort of triggered or dovetailed with the search for stem cells in the intestine. And the first stem cells type to be identified from Hans Klaver's group a number of years ago now were the crypt-based columnar cells. And these CBCs are highly proliferative. What you're looking at is a crypt structure of the intestine. And this is the single cell layer mucosa. The CBCs are these red cells down at the bottom of the crypt, highly proliferative. Here are a couple of biomarkers. They grow, divide, and give rise to daughter cells or transit amplifying cells that are just as proliferative. And these are like teenagers. They are, very active, trying to decide what they're going to do or turn into or become, and they leave the niche and they move up the conveyor belt of cells here to differentiate into mature cell types. The CBCs are heavily dependent on Wnt. The Wnt are provided by neighboring cells, fibroblasts, and these neighboring cells. They're for all of the gene programs that I just listed. There is another stem cell pool in the crypt, not as well studied because it's only been recent. We've had the technologies to identify them. And these are variously called +4 cells, reserve or revival cells. I'm going to call them regenerative stem cells just to be politically diplomatic for each group. But these cells are not proliferative. They're quiescent, and they stay that way as a rare population until there's injury in the gut. And that inflammation and injury triggers these regenerative stem cells to come out of quiescence and begin proliferating. CBC cells are very fragile. Any kind of injury kills them and they slough off, but regenerative stem cells can proliferate and move down and turn into CBCs and replace them. They can also move up this way and turn into TA cells. And the performance objective of these regenerative stem cells is to rebuild a tissue. So the origin of colorectal cancer has been understood for many years due to some transgenic mouse experiments that Hans Klaver's has done, is to be the CBCs, because they could introduce mutations in these stem cells and show that they created adenocarcinomas. But that is being sort of revisited for the following reason, and that is that if 80 to 85% of patients have a colorectal cancer, transcriptomically and genomically, those cancers can be classified into different subtypes. If they all come from the same stem cell, you'd think that they would be a little bit more uniform. So recently, 2 papers have come out on a reconsideration of the classification of colorectal cancer, which previously had had about 5 or 6 subtypes. One is from Jeffrey Rana, who's at University of Toronto, right where you guys are right now. And the other is from Owen Sansom in Scotland, the Beatson Cancer Institute, and his colleague Simon Leedham at Oxford. And they said, basically, there's only 2 types of colorectal cancer that have Wnt mutations. In Simon's case, they said the tumors just vary in their prevalence of cancer cells that look like CBCs or RSCs. In the Jeffrey Rana's classification, he said, forget about Wnt, you can classify colorectal cancers into 2 types based on this kind of YAP pathway, which is the hippo signaling pathway, which I'm not going to describe. What I'll show you towards in this talk is that we believe this binary classification is quite real. But step back a minute and show you the data that first triggered a collaboration with John. You can see these tissue slices histochemically stained for the prevalence of beta-catenin, so a biomarker of Wnt signaling, and a biomarker of glycolysis. We had found that Wnt signaling regulates a glycolytic metabolism program. And these are tumors created by injecting a human colorectal cancer cell line into mice and growing them subcutaneously. And you can see this spotted pattern, I'm going to blow it up for a little bit, where you see clusters of cells that have very high beta-catenin, surrounded by cells that have lower levels, and the same is true for this biomarker of glycolysis. And I showed this to John and I said, is this some kind of data? I don't understand this. I've never seen this pattern before. Is this something that would be of interest to a mathematician? And he said, absolutely yes, and began to talk about reaction diffusion principles and Turing models. And in fact, Wnt signaling, because of the Wnt ligand having such a short range of diffusion and having the ability to turn on inhibitors that have a longer range of diffusion, this signal transduction pathway lends itself to reaction diffusion structures. And so I'm going to cut to the long chase because this is published. When Mary and John went to the drawing board and started to model the patterns that we had seen in these xenograft tumors. I'm just showing you a snapshot from the paper where, you know, cells with high Wnt activity are autoregulatory, and they turn on the expression of a Wnt inhibitor that diffuses further. And then there's similar kinds of descriptions of this switch in metabolism that we had identified, driven by Wnt signaling. And this model was used for MATLAB simulations. So Mary could show that either 2D or 3D, if you simulated the growth of tumors with this reaction diffusion set of relationships, that the spotted pattern for Wnt and glycolysis emerged with parameters that really resemble what we saw in tumors in terms of the size of the spots and their arrangement. And this is part of this paper that we published together. And my student George put together a graphic that the editors chose for the cover of the journal. It was very, very fun to get this paper out there. Now, I learned that models are only useful if they make predictions, sometimes, you know, counterintuitive predictions that biologists can go test. And in this study, what John and Mary predicted were a couple of things. One, that if there was a magic inhibitor that could effectively inhibit Wnt in tumors, that would trigger an increase in the diffusion range of Wnt ligands that might extend their reach. And in fact, what George found when he did RNA-seq and harvested these tumors in an RNA-seq, we could, sorry, let me back up. We created tumors and used a genetic trick to mimic inhibition of Wnt signaling using some dominant negative constructs of the left TCFs. And what we found in bulk RNA-seq of those tumors that had Wnt inhibited, that there was a very sharp increase in the expression of these secreted frizzled related proteins. These are the things that bind to Wnt and extend their range. And this was very paradoxical, but it turned out that the prediction that John and Mary had made was true. And the second prediction that they made, or a similar prediction, that we looked in patients. George was able to get a data set from colorectal cancer patients that had been treated both with radiation and chemotherapy. And diving into this data set, he found that when the tumors were attacked by chemotherapy and radiation, Wnt target gene expression declined sharply, hinting that Wnt signaling had been inhibited. And interestingly enough, in those same tumors, this secreted frizzled protein expression went up 5 to 50-fold. It was very dramatic. Again, we weren't able to show that Wnt ligands could extend their diffusion range, but certainly the players that enable Wnt's to diffuse further were dramatically increased. And finally, another prediction Mary and John made was that if you inhibited both the activity of this enzyme in glycolysis and Wnt signaling together, rather than just, you know, this problematic extending of the range of Wnt ligands, you would inhibit tumor growth. And George was able to test this in vitro with tumor spheroids growing in fibrin, using an inhibitor of this pyruvate dehydrogenase kinase, a glycolytic enzyme, and an inhibitor of Wnt signaling. They synergistically suppressed colony tumor sphere formation. I'm not showing the data here in the interest of time. Now, this was all very exciting, and but the fact that we had this heterogeneity and that was patterned in tumors, that we could model it, prompted us to start to learn how to do single cell RNA sequencing. And this is the ongoing collaboration with John that we have going. It was interrupted by COVID, but so it's taking a lot longer than we thought. Nevertheless, this is where my student Lindsay Hosahama came into play, and also two postdocs of John's, Wamin Yang and Shushang Wang. And I don't have pictures of them, unfortunately, but I do want to acknowledge all of their wonderful modeling work that they did with Lindsay. So what you're seeing is the xenograft tumor I showed you before. And this is a UMAP showing single cell sequencing where every dot is a cell. And what we could see right away, and this is illustrated in the heat map, is that in this xenograft tumor with spots, there were two very different cancer cells. So here we're just calling them subtype 1 and subtype 2 until we could characterize them further. I'm going to skip over a whole bunch of work that took Lindsay a very long time. But the bottom line is that subtype 1 and subtype 2 are two cancer stem cell subtypes. You're seeing a phase contrast image of one subtype here that's called rounded. These are not single cells. These are spheroids, so about 50 cells or 100 cells, loosely attached to the dish, very tightly adherent with each other. And these look like transcriptomically like the CBCs, the crypt-based columnar cells, because here's the biomarkers LGR5 and MYC and others. And they do not have this pathway YAP hippo signaling active, but these are biomarkers of CBCs. The other cell type in this mixture was very adherent to the culture dish, sat down like a fried, it looks like a fried egg, so we called it adherent. And the gene expression signatures look very much like the regenerative stem cells, a fetal intestine gene program. This YAP hippo pathway is very active. The reason Lindsay could separately culture and characterize these cells is from the single cell RNA-seq data, she found ways to flow sort and separately culture these cells. These subtypes are from the same patient. They are from the same tumor because we did STR profiling and we sequenced the genomes of these cells. Yet they couldn't look and behave more different from each other. And transcriptomically, as I said, they're very similar to normal counterparts that you naturally find in the crypt. They also sort of support this proposal that there's a binary classification of tumors in colorectal cancer that I described before, because they differ by YAP signaling and they match these two subtypes. So that's very interesting. Now, John's postdocs, Wamin and Shushang, took this single cell data. Here I'm showing you another data set, and using the transcriptome signatures could identify them, giving them slightly different names. So now the CBCs, crypt-based columnar cells, are called colon or cancer stem cells. And the adherent RSCs, the regenerative stem cells, a larger population in this tumor, were actually the Wnt highs, and then had very strong signatures of Wnt signaling. Again, paradoxical. Because we also had other cell types, we could also identify immune cells and stromal cells. And sorry, Paul, I don't have time to go into them, but they are extremely interesting. And this study is now that Lindsay has characterized the tumor microenvironment in these tumors. It's all up on bioRxiv, and you can access this to look at the interesting findings that we found. And I'll get back to that towards the end of the talk. So taking this model and again, using reaction diffusion principles where, Wnt high has a feed forward and then also negative feedback with this secreted, here now we're showing DKK4 because it's expressed very highly in these cells, inhibitor, we just set up a pattern of reaction diffusion, but also based on the transcription data from the single cell, other signaling pathways such as FGF, and EGF, not shown here, and bone morphogenic protein, BMP, were shown to be very important because the receptors for these signals are on these cell types. And also from the stroma, Wnt ligands and FGF ligands are highly expressed. Using this sort of relationship, a preliminary model was set up where in silico growth of tumors, again, in a 3D character, again, recreated that cancer stem cell heterogeneity that we observed in tumors. And in this in silico growth, recapitulated the relative abundance of the different cell types that were observed in the single cell data set. Sort of suggesting that this model, even though it's early stages, is approaching what we're observing at the single cell level. Now I'm going to sort of end here with one vignette story again of how a model, if it's good, will make a prediction that biologists will test. And in this case, very early on, Waterman said that in this model that they've put together, these bidirectional arrows suggest that these stem cell subtypes must switch, that cancer stems, that the CSCs might switch to Wnt, et cetera, based on their location, metabolic and microenvironment around them. And for years, like 3 years, Lindsay tried very hard to test this prediction using her purified cultures, by mixing them in culture, by putting them as mixed populations in tumors. And in every single case, there was no robust result that said, yes, that switching between these cell states was triggered. There could be small hints, but there was no real strong evidence that this component of the model was really true. Until we started to look at these papers that are proposing a binary classification. So here I'm showing you a abstract from the Jeffrey Rana paper, suggesting that there are two subtypes of tumors, YAP-on and YAP-off. And in that paper, they said if there are these two types of tumor subtypes, they should have different vulnerabilities for different chemotherapeutic agents. And in the YAP-on subtype, which is this adherent subtype, let me go back a minute, these guys, they would be vulnerable to inhibitors of Src family kinase signaling. So FGF signaling, EGF signaling, BRAF, and even, and this gets back to Vivek's talk, components of the extracellular matrix. And such that if you were to treat these tumors with these inhibitors, that would be an effective therapy for patients that have that kind of tumor. Whereas in the other kind of tumor, perhaps based on this rounded CBC subtype, inhibitors that attacked metabolism or the oncogene MYC would be more effective. And so Lindsay was testing this particular prediction from the paper. Let me just show you that the reason they predicted this kind of tyrosine kinase inhibitor treatment is because the Hippo-YAP pathway through Src family kinases is very tightly linked to these tyrosine kinase receptors. And if you interfere with the activity of these kinases, you really heavily influence the activity of EGF, FGF signaling, et cetera. So Lindsay took the cells, the adherent cells that have a lot of YAP Hippo signaling and should be vulnerable to Src family kinase inhibitors. And she chose the Src family kinase inhibitor dasatinib because it is approved for therapy, a second-line therapy in colorectal cancer patients, and treated it. And shockingly, within 1 day, 24 hours at different doses, and then fully at 3 days, she found that the morphologic phenotype of the adherent cells converted into what looked like rounded cells, sort of right before her eyes. And this was the first hint that something that interfered with the signal transduction networks that were set up in these adherent cells actually could flip them, switch them into rounded looking cells. She harvested these cells and she did RNA-seq on them to see what transcriptomically was changing. And this is just crude. We're doing a lot more analysis of this. What she found is that genes that are highly and uniquely expressed in the adherent cells were the top most downregulated genes, and genes that are, uniquely and biomarkers of rounded cells were the top upregulated genes, confirming at the transcriptome level that interference with YAP signaling is causing switching. And therefore, the reason that she wasn't seeing switching in vitro in her cultures or in simple xenografting must be that the environment isn't strong enough to unlock the cells that are in this signal transduction network. Why is this important? Why are we belaboring this? Well, as I told you, dasatinib is a chemotherapeutic agent. And so it's a Src family kinase inhibitor. And the prediction from the Jeffrey Rana paper is that it would be an effective therapy for these tumors. In fact, what we're seeing is that it switches the cells, doesn't kill them. It switches their stemness to a different stem cell type. And this is important, and this is my last data slide. I want to show you what happens when we inject these two different stem cells into the wall of the mouse colon to ask how the tumors grow. Here's a normal colon that's been sort of rolled up like a rug, and you can see the dark purple mucosa and the underlying pink muscle layer. And we inject tumor cells in between the mucosa and the muscle. When we inject these fetal-like YAP-active adherent cells, they grow tumors, but they're very well bounded, as the dotted line suggests. They grow in between the mucosa and the pink muscle layer. When we do the same thing with the CBCs, which are highly proliferative, the tumors are terrible. They invade into the muscle, as you can see here. They destroy and invade the mucosa. They plug up the lymphatics, so there's all this kind of edema. This is a highly, highly aggressive tumor type that would have a poor prognosis in a patient. And that is precisely the type that this Sarc family kinase inhibitor sunitinib converts the adherent cells into here. So it's a cautionary note that simple treatment with the Sarc family kinase inhibitor for patients that have this kind of tumor type might actually convert those tumors and make them more aggressive and increase patient mortality. So I'm out of time. I'm going to stop there and tell you that this is an ongoing collaboration with John, even though it will now be a distant one, geographically a separated one. I want to acknowledge all of the work of John and his group, Mary Lee, Wamang Yang, and Shushan Wang, as well as my students, Lindsay Hosahama and George. And with that, I want to thank you, and I'll take any questions.
Thank you very much for just a beautiful talk. I want to say, we have a lot of hands up, and we also have a hands up in the virtual space. So I want to maybe move on here.